G'day everybody, we've got Keith Wiley here, who's a fellow of the Brain Preservation Foundation, as well as the author of Mind Uploading, Taxonomy and Metaphysics of Mind Uploading, So welcome back to the channel there, Keith. So we're going to be talking about the Brain Preservation Prize, which has recently been won. So yes. So starting off with a 1,000-foot view, do you want to just give us a brief introduction to your interest in this field?
Right. So the Brain Preservation Foundation is sort of tangentially related to my interest in mind uploading. I've discovered them after I wrote my book and formed a sort of a working relationship with John Smart and Kenneth Hayworth, who run the foundation. And the connection is that brain preservation is seen as well, for one thing, kind of a bridge to the future. It's a way of sort of putting yourself into suspension to await the decades until mind uploading is possible. Alternatively, some of the ways of achieving mind uploading that are all sort of on paper right now actually start out with a stage that looks very much like brain preservation. One of the initial stages of mind uploading in one of the more popular methods consists of essentially putting the brain into a solid stasis and then slicing up the brain and scanning those slices. So in that sense, brain preservation is basically just the first step of a futuristic uploading procedure that might be developed someday.
Excellent. And I should say that I've interviewed John Smart, I think back in 2010 when he was in Australia. That was a great experience. He's a really nice guy, I found. And he also, I did an interview with him about Future Day. And Kenneth Hayworth as well. I did a classic 4-hour marathon of an interview with Kenneth Hayworth about his views on brain preservation, cryonics, and uploading in general. And I like his bent that what we're trying to do, or what you guys are trying to do, is have scientific existence proved before promising too much.
Yes.
And I found that's important, especially if you want to convince the scientific crowd. And
exactly.
Part of convincing the wider audience is convincing the scientists that it's a tractable problem that can be solved.
Yes.
And then, other people will start listening once there's scientific credibility behind the ideas. I think philosophically the ideas have been working for quite some time, but yes, it's nice to have some existent proof there, and hence the need for the Brain Preservation Prize, or the utility thereof.
Yes, the Brain Preservation Foundation was initiated to start with 2 cash prizes for 2 different stage accomplishments. Kind of in the spirit of other sort of cash prizes for scientific or technological achievements. I think the famous progenitor was the Lindbergh Prize for flying across the Atlantic. There have been X prizes and Google prizes, and there are a bunch of similar ideas. So what the Brain Preservation Foundation did when it was founded was it motivated with 2 cash prizes progress into sort of advancing research of how to preserve a brain in a way that satisfies the scientific community, that the critical parts of the brain have been preserved, those parts that would preserve a person's cognitive features. So basically, what neurological features do we need to preserve in order to be confident that we have preserved the important cognitive features, and the important cognitive features are then memories. So basically, first of all, what is the consensus of the neuroscience professional community on how memories are encoded? And then what would be a stasis or preservation procedure which would satisfy the professional community that the neural encodings of memory are actually being successfully preserved? So the BPF started out with 2 cash prizes, one the small prize and the large prize, intended, not larger prizes, but the prize for preserving a smaller brain, a rabbit brain, and then a later prize for preserving a pig brain. And essentially, if a researcher could demonstrate to certain sort of thresholds of rigor, which I'll go over in a minute, that they had accomplished this task, then they win the cash prize. So what are those thresholds? Well, first of all, we really want the professional neuroscience community to sign off on this. So one popular sort of a threshold of credibility in the academic and scientific communities is the process of peer review publication. So in order to win the BPF prizes, it's not enough to just sort of put out editorials and news articles or make a splashy video. You have to successfully get your research peer-reviewed and published in an established and respected journal. And I, off the top of my head, I actually don't remember what that journal was, so I'm not going to speculate. But the winning team did do that. And then, in addition to publication, they had to submit brains that had been preserved according to their attempted procedure for independent analysis, and that analysis was performed by Kenneth Hayworth of the BPF. So you can imagine that he's going to demand very good quality because he's the one handing out the money. He's not going to give out the money unless he himself is actually satisfied. So it's sort of good that the person in charge of giving away the money is the one you have to satisfy if you want his money. So that's a pretty good sort of rigor there, and that's what was accomplished in 2016. Robert McIntyre's team at 21st Century Medicine preserved a rabbit brain, submitted it, and Ken took electron microscopy sort of images of the ultrastructure and investigated those at the microscopic scale, and showed those images to other professional neuroscientists, and they all came to a consensus that this rabbit's brain had been preserved in a way that preserved the critical neural features of that rabbit's memories. And then in 2018, just this week, it was announced that a larger, more complex brain, a pig brain, had been preserved in the same manner. And it's no huge surprise, of course, that this research, having built on the previous research, was won by the same team. McIntyre's team, again, was the winner of this second prize as well. So that is what this week's big news was. There was a fair amount of press release about it, and we would like the public, and we would like the professional and academic neuroscience communities to keep carrying this forward at this point.
Well, what aspects of this do you think appeals to people? I mean, when you there was another one. There was the small brain, a rabbit brain, which was preserved a little while ago. What's the extra, what extra has been achieved? What are the extra bonuses that we've achieved from preserving a larger brain, which arguably, I guess, is harder?
Yeah, well, I think I'm actually not an expert in sort of the subtle details of these procedures, but I have a pretty solid grasp of it, having sat in on the organization's meetings for several years now. One of the crucially difficult parts of preserving organic tissue is perfusing that tissue with the various chemicals that are used to preserve it. So you can imagine that the more volumetric and literally the thicker or wider a chunk of tissue is, the harder it is to just sort of diffuse chemicals through it in a way that you're satisfied you've done so effectively and gotten it to every cubic millimeter of the entire tissue. So in that sense, a bigger brain's harder just because there's deeper tissues that you've got to get the chemicals into. Another hard part is that this wasn't a requirement for the prize, but the method that won the prize is a temperature-based method. It uses low temperatures for the preservation. Everybody knows that low temperatures keep things from spoiling. That's why we put food in a refrigerator. So the point is that if you're going to use temperature as your technique, then you again have a problem from the thickness of your tissue, because the tissue is holding heat. It's actually hard. It just takes longer to cool a wider, thicker chunk of tissue down because it's holding heat at its center. So for all these problems, if you can demonstrate that you can preserve a larger brain, you've demonstrated that the technique, the procedure, it really demonstrably works to a higher level of satisfaction. I suppose the interesting question is, okay, so we did a rabbit brain, we did a pig brain. How does this prove that we can preserve a human brain? Would be the follow-up question. And I think, again, now I'm not speaking for anyone but myself at this point, but I think it doesn't prove it. What it proves is that there is a technique now, and it was the same technique. So we're on a ramp here from rabbit to pig. There's a technique here which seems to be now demonstrated to be able to do this if we just work through the details. Basically, a human brain can probably be preserved in essentially the same way as a pig brain. There's no particular reason to my understanding, based on essentially on Ken's description of everything and Robert's description. In their opinion, a pig brain is large enough to demonstrate that a human brain is now within reach of this procedure. So the procedure should be able to do this.
Okay, so I mean, if someone were to then be faced with the option of different techniques to choose between in order to, like, okay, someone wanted to get cryonics, and they thought, okay, well, getting deep freeze or doing something similar to maybe what Alcor Foundation or other organizations might do compared to what's been going on. I'm not sure if it's plastination. Is that similar to what the technique that?
No. Right. Right. So there's a few questions there. What is plastination is one question, but another question is, how does this new procedure that McIntyre's team used, called Aldehyde-Stabilized Cryopreservation, ASC, how is it different from?
That's not plastination. It's what was previously described by John Smart as an alternative to cryopreservation.
Right. Sean McCullough and others have looked into what are essentially room temperature preservation. They don't rely on low temperatures. They rely on, again, some chemical process that I'm certainly not particularly knowledgeable of, that would turn the brain into, I'm not sure if it literally turns it into, chemically what would be called a plastic, or if plastination is more of a verb, sort of some sort of process plastinates things. I'm not entirely clear on that. But there was this other procedure that could actually preserve things at room temperature. You could put a preserved brain on your bookshelf for 3,000 years. It would have advantages, because cryopreservation is a process that has to be maintained. You don't stick something to cryopreservation and walk away from it, because these low temperature substances, nitrogen and whatever, they actually evaporate away. So someone has to sort of watch the facility and keep pouring in more nitrogen into the top of the tank or something. So a room temperature technique would be fabulous. And I'm not sure where that will go. It's quite possible that additional preservation methods will come along. They didn't win these prizes, but the research is ongoing. But for now, what we have demonstrated and working is this ASC method. So how does that method differ from? We know that the cryogenic process has been used by the cryonic industry. Those are slightly different words. Cryonics is essentially the business or industry or application of the cryogenic, sort of method to preserve. When applied to preserving people, you call that cryonics. How does that differ? Well, first of all, I'm a little reserved in where I'm critical here, because I'm obviously speaking not only on my personal behalf, but on behalf of the organization. So the organization has its own sort of official positions on this. But cryonics, as we all know, whether or not we wish this was the case, has not gained widespread credibility in our society. There are people who are on board with it, but there are a lot of people who are not, and there are a lot of scientists who are not. That means that, well, for one thing, what if it's not actually working very well? Then basically what that means is we don't really have a good way to preserve people who would like to have the opportunity to sort of skip forward in time. But even if it did work, let's say that cryonics in its sort of not particularly well respected way, but what if everybody who doesn't like cryonics was wrong? What if cryonics actually worked? It would still be an unfortunate situation, because very few people are on board with it. So no one's really benefiting from it, even if it works, because we don't take it seriously enough. And I think we are right to not take it seriously yet, because I'm a big believer in really careful, rigorous, scientific, not only processes, but also the overarching peer review and checks and balances built into how our scientific institutions work. This is how we make sure that things work.
Well, I agree. From a scientific point of view, it has not yet, as far as I understand, got the credibility, and shouldn't get the credibility that that we'd like it to, just by being a great idea without the background of being tested, peer reviewed, and replication tested, and having all sorts of empirical studies being done to support it. And it shouldn't be given scientific credibility until that's the case. However, it does seem philosophically and ethically like a great idea if preserving life is a worthy standard. And also personally, if we do want to see the future, if we do want to see what's in store for humanity in the 100, 1,000, 20,000 years, and what the future of life might be like, and it seems like it's one of the least desirable options to be cryopreserved, but on balance, oblivion seems a lot worse, and maybe being woken up into a world full of suffering may be worse as well. But I guess, if you were to rank all the possible outcomes, of course, staying alive would be a lot better than dying and being cryopreserved.
It's amazing how many people don't agree with you. A lot of people do seem to be against this, and they're not necessarily against it because they think cryonics is goofy or something. They're against it. People convince themselves of ethical arguments against it, which is frustrating because we at the BPF think that at the very least it should be sort of an individual option, that whether or not any one person agrees with it, they shouldn't have such a strong ethical position against it that it would be prohibited from others. But a lot of people actually do think that, and we're going to have to really grind through this now that the viability of putting people into stasis for long durations of time with the potential of reviving them in the future, which we need to talk about how revival works in this process, because that's a big issue. Our society is going to have to make some decisions about this, because right now cryonics is a small community and most people don't take it too seriously. But if an improved cryonics method came along and became widespread and cheap and accessible, and not just the subject of sort of accusations of elitism, but actually became standard practice in hospitals, we could be potentially looking at thousands, even millions, even tens of millions of people who would have access to this. And now, which could be just a few decades from now, our society is really going to have to decide whether, collectively, we're going to allow people, the individual right and individual self-actualization to make this decision for themselves, or are we going to, as a society, come down on some sort of ethical judgment that actually prohibits this, that people aren't allowed to do this, not only because they wouldn't choose it for themselves, but because other people decide that it won't be allowed. And I think that would be that would be tragic. But society has made sort of bad decisions for everybody in the past, and this is why we should start having this conversation now, so that we have literally decades to figure it out.
Exactly. And my opinion is, yes, if we put a moratorium, or we to halt this sort of research in fear that it might be used for the wrong reasons, or it could be unethical to keep people alive, even if they wanted to keep people alive for long periods of time. We're not there yet, and to assume that we've discovered everything we need to know about the ethics and how it plays out in the future now, I think it's incredibly naive. We don't know what the future will be like. We don't know what other technologies that we'll have in order to reduce, I guess, environmental impact of people on the Earth, increase propensities for people's flourishing and happiness, and reduce suffering, such that lives are really, really much worth more living than they may be even today for everybody. These are all possibilities, and the way that the future plays out, I guess, that's contingent on whether or not, such a technology like cryopreservation or reanimating a mind is ethical. And I think we shouldn't be so naive and so sure of ourselves to have come to the right ethical conclusion today to justify stopping the research, or to shun it, or to quell it, or
that's a very good way to put it. That's a very good way to put it. Right now, all we should do is continue to research it. Absolutely. So one thing we should definitely talk about is the potential methods of revival. Conventional cryonics, the method by which conventional cryonics is applied, whether or not you are sort of buy into its technical viability, or whether you think it's just killing people, wherever you come down on that, at least the potential revival method is kind of what we often see in science fiction. The great Woody Allen movie Sleeper, but then Futurama, and really almost an uncountable number of science fiction examples show a similar path to revival, which could be colloquially described as thawing them out. Basically, they are biologically sort of revived. And now one caveat here is that the cryonics companies do often offer a cheaper option where they will only preserve your head. So one might ask, why would you want to preserve your head? My cat is knocking things down. I'm sorry.
Cool cat, man.
So it's interesting then. Cryonics people, the current sort of community, seem to, I think, prefer what's called bodily revival. They want their bodies back. They want their physical brains back, but some of them are on board with the idea of a cloned or robotic body. They really think that at most it is their brain or perhaps their head that must be crucially revived, but the body is optional. And we can see good evidence of this all around us, because it would be somewhat disingenuous to amputees to suggest otherwise. Clearly humans are well capable of forming their sort of self-percept of what they are as a person in the absence of limbs and organs, and in the presence of prosthetic and replacement organs. We incorporate these additional or replacement modalities into our sense of identity quite smoothly. So that would explain why many people are sort of limit their bodily requirements of identity to at the most their brain. So the new method that won the prize, that preserved a rabbit in 2016 and now a pig this week in 2018, ASC. The way that it achieves a vastly superior and higher quality preservation is biologically irreversible. It begins with a stage of glutaraldehyde perfusion throughout the brain. Glutaraldehyde has been used for decades to prepare tissues for analysis in labs. It has never been used, sort of ostensibly to preserve, something to be, much less a brain, to be revived later and wake up. But it has been used to preserve tissues. It's kind of like using formaldehyde. The thing about glutaraldehyde is the very way in which it preserves tissue is also makes it sort of impossible to get it back out again. It creates these very tight molecular cross-link bonds that we really have no idea how you would ever undo that. So first of all, why does ASC use the glutaraldehyde? Because glutaraldehyde accomplishes a very high quality room temperature preservation. This room temperature preservation would not be good for centuries or millennia. So you go ahead and lower the temperature anyway, and you use this low temperature liquid nitrogen technique. But what the glutaraldehyde buys you is weeks, literally weeks, over which to very slowly, very carefully lower the temperature, because it is now preserved in glutaraldehyde, which is a good preservation for several weeks. No decay will occur over that sort of span of time. So you can lower the temperature very slowly. You can even bring the temperature back up for a few days and check some part of the brain that looks like it was having trouble, and then bring it back down again. You have weeks to get it down and make sure that it's a really high quality preservation. The current way in which sort of the cryonics industry attempts to get a body or a brain to low temperature has to do so in hours, because the brain is decaying immediately, because they don't use any glutaraldehyde like upfront preservation. So anything that remains warm is decaying, so they get it down to temperature really fast. And that rapid process is where most of the problems, if there are problems, seem to arise. So at any rate, Well, so then that begs the question: how is ASC performing any sort of preservation that would lead to future revival if I'm admitting that you can't biologically revive brains that are preserved in this manner? And at this point in the discussion, we take a sharp left turn into metaphysical philosophy.
That's right.
Where
aboard the ship of Theseus.
Yes. So Theseus, not Prometheus. So the question is,
yes,
what is it that represents yourself, your mind, your identity? What is it about you that makes you alive or conscious, or this thing that is even more abstract than consciousness, which is personal identity? What makes you you instead of someone else, if that statement even means something? This is a question that philosophers, metaphysical philosophers, have been, sort of tackling since, I think, Socrates, certainly Plato. And there are several theories on the nature of identity. Derek Parfit's book, Reasons and Persons, is one of the good places to start if someone wants to read about this. But in short, what are the various theories that have been put forward? So one of the most popular is called body identity, which we should actually start calling it something else in this day and age, because we should call it matter identity, because it doesn't really mean you need your body. I mean, as I just said, even people who want their physical brains back will often, are okay with only preserving their heads. So when I say body identity, what I really mean is material or matter identity. This theory says that your identity is, closely tied to the specific set of atoms, basically, that make up your brain.
Atoms, right?
Well,
no, pattern identity, right? No.
Well, pattern identity is a different one. Right now, I'm describing body identity, and in body identity, you really think that your atoms are who you are, and this is why many people really want themselves to want their own brains to be the revived entity, that if you came back in any other form, which I'll get to in a moment, it wouldn't preserve your identity. Instead, they feel that they would die anyway. And even if the process seemed to technically work, some other new person would sort of, materialize out of nothing and to take their place. So what are the other theories? One of the most popular or most widespread alternatives is called psychological identity. In psychological identity, your cognitive features define who you are. And by cognitive features, I really basically mean your memories. It all comes basically down to your memories. You can say it's your personality. You can say it's a variety of things. But what it really comes down to is the memories that indicate your entire life history, which then is where your personality comes from. And it's also where your contiguous sort of memory of yourself over your past is just a function of your memories. So psychological identity says the memories are what indicate who you are. So if you can preserve the memories, you have preserved the identity. Well, can we preserve our identity? Can we preserve our memories without actually being dependent on some particular set of matter? And of course, this is a popular trope in science fiction now, where you sort of basically ponder some thought experiment, which consists of taking the way in which memories are encoded in the physical brain and replicating that pattern of memory encoding in some other system. And canonically, it's a computer of some sort, or potentially a sort of a futuristic version of what computers will turn into as technology continues to evolve. So the idea here is psychological identity then becomes closely tied to pattern identity, where who you are identified as, is indicated by the physical pattern of your brain. But again, it's not really the pattern of the atoms that matters. It's the fact that those patterns underlie memories. So the way in which we believe that memories are encoded in the brain is essentially this big, vast network, which is called the connectome, which is the way in which the billions of neurons and trillions of synapses all connect to each other. So at this point, we're sort of abstracting away from the brain. It's not really about atoms. It's not really about molecules. It's barely about cells. The neurons are cells. What we're really getting at is this abstract notion of a network. And it's that network and particular critical properties of the network, properties of the way in which the connections actually operate, in which they communicate information between the neurons. This informational description of what's going on in the brain now becomes the salient information that indicates who we are. It's the structure and properties of the information passing and information processing network that becomes your identity. So once we are at that level of abstraction, we can really start thinking about other things we can do, such as getting away from the brain, or changes we can make to the brain. You know, once we're not dependent on components of the brain, we can replace small pieces of it. Ted Berger is working on a hippocampal prosthesis that helps people. There are people who, many people who have deficits in their hippocampus such that they cannot convert short-term memories into long-term memories. And his prosthesis is an early stage research proof of concept that we can put essentially a signal processing computer-like prosthesis in place of a damaged part of the brain that takes the incoming neural signals from one part of the brain, processes them in the way that the damaged part of the brain would have, and outputs the same output signal that damaged part would have. And that supplantation, that supplanting that part of the brain actually works. He's been able to demonstrate effectively repairing the ability to form long-term memories. So this is all very early. It's only 2018. But what is the extension then? If we can replace more and more and more parts of the brain with a computational component, then that implies that the brain was never really the important part. The information was the important part. The pattern was the important part. The atoms didn't matter. Even the neurons didn't. The fact that the neurons were biological was never really the important part. If it was, then things like Berger's prosthesis would not work. It's the much more abstracted information processing aspect of it that is making the brain produce mind-like qualities.
Well, so at this juncture, just quickly, I think some people would argue that aspects of the brain may be like, you know, the raw fields or the consciousness may turn out to be dependent on like a substrate more complex than silicon, like biological or carbon-based substrates, but it doesn't mean that in the future we won't ever achieve computation, artificial computation on such types of substrates at all. So just because there are some concerns out there, I'm just bringing that up.
Right. We don't know for certain, of course, exactly either you know, how the brain works in general or how the brain performs certain aspects, especially these sort of complex questions of how the brain inspires these kind of metaphysical properties like qualia and mind and consciousness. Whether or not it depends on some sort of biological thing that cannot be you know, sort of reproduced by a non-biological material or trait. I suppose that's an open question, but one of the prevailing theories right now is that it is an information processing task. So if you know, if that theory is the correct theory, then none of the biology really matters. The only thing that matters is transmitting and processing signals the same way. So if that is the correct theory, then we don't need ribosomes and proteins. We just need to transmit signals around a network in a similar fashion to the way that the brain does it. Now let me bring this all home. What does this have to do with ASC preservation? So like I said, ASC preservation uses this glutaraldehyde step, which can't be reversed. So if the whole sort of theory that I've set up in advance here, this whole pattern identity theory, is a reasonable way to conceive of how our minds work and how our metaphysical identity is indicated, right? Then the idea is to take the physical pattern encoded in a preserved brain and scan out that pattern and reproduce it in a computer, and this would be called a whole brain emulation. It would be an emulation of how a particular brain processed information. And we even have decent early stage designs of how this process would work. Most likely you would slice the brain up into very thin slices and scan the slices. And there are different approaches to this technique. You can physically slice it and then scan the slices one slice at a time, or you can what's called block face scanning, where you basically take a picture of sort of the surface, and then you essentially vaporize away, you know, a few atoms worth of the brain, and well, it might be more than atoms. You vaporize away some thin top of the layer, and then you take a picture of the surface again. So it all comes down to converting the three-dimensional sort of volumetric structure into two-dimensional surfaces, which then can be imaged. And what you do then, most viewers are probably already familiar with this, just because it sort of comes up a lot in our society. But so from these two-dimensional images, you then infer the three-dimensional network structure that had been in there in the first place. You can sort of imagine if you took, I'm trying to think of an example. If you took like a ball of yarn, right? So just a big snarled up ball of yarn, right? You could imagine that if that ball of yarn was frozen solid, so that it wasn't going to be all wiggly, right? If you froze it absolutely solid and then sliced it very thinly and kept track of the slices and kept track of how the slices aligned, then you could actually build up sort of a computerized 3D model of that snarled up ball of yarn. So you would reconstruct in software the three-dimensional structure that had been this sort of wiggly-waggly one-dimensional ball of yarn. Wiggly-waggly is an official term, by the way. So that's the idea with the brain. We want to capture that connectome, the network of neurons connected at the synapses between neurons, capture whatever other chemical properties are required. The glutaraldehyde and cryopreservation will lock all the particular molecules in place, so we can figure out which kinds of molecules are where during our scanning process. And put all of this into a computer model, which would be called a whole brain emulation. And when you run the emulation, you are then running a computer model of that brain. And you know, all of this sounds very futuristic. Well, guess what? It is futuristic. This isn't something we're going to do now. I don't know if we will do it this century. Some people are much more optimistic than that, but I tend to be pretty sort of reserved about these sorts of things. I don't really care if it's done in this century. I think, and I don't really care to have the idea downplayed because people think that the prognostications are too optimistic. To me, it's not interesting whether or not it happens this century. To me, what's interesting is that it is technically, physically sound to theorize about this and to speculate that, you know, barring a societal collapse, we will get there in time. That's sort of where I stop as far as wild speculations are concerned. I just think that inevitably technology will be able to do this at some point. And then what are the philosophical implications? If you run a whole brain emulation and you hold a theory of identity that is based on pattern identity, and not everyone does, but if you do, then your interpretation of a whole brain emulation is that it preserves the original person's identity. They have survived into this emulation. And the word for interpreting the whole brain emulation as a preservation of the same identity is mind uploading. That's what we call mind uploading.
I guess in the near term, if we did have an existence proof of a mind, like, you know, a rabbit or a pig or some other animal transferred into a virtual world, or, you know, existing, then that would be a pretty strong existence proof of it working for possibly for humans as well. And then you'd probably see a whole heap of money funneled into the idea, into the research program. And that would be good in the near term, I guess.
Right.
But yeah, that's just I didn't want to break the continuity of yours.
So there's a few questions there. First of all, emulating, we might think, okay, emulating humans is hard, so we'll just emulate a rabbit first. Rabbits are hard. Right now, the only thing we've ever managed to create any sort of an emulation of is a C. elegans roundworm, which consists of, I think, about 900 cells total, and maybe 300 neurons, or I might be way off. It's comedically minuscule. There are designs for projects that will attempt to do things like this with Drosophila, the fruit fly, and things like that. So we're in very, very early stages. We're not going to be scanning or capturing the connectome of a, first of all, aside from whether we could run the emulation, we can't get the scan yet. We don't actually have the technology to fully scan and deduce the connectome of a brain as big as a rabbit's or a mouse or anything like that quite yet. But then even if we could, we really don't have computers that are capable of it. There are a variety of approaches to how to design computers. If you take the way all computers that we think of as computers work, the one on your desk, the one on your phone, these are von Neumann architectures. And the only way we can make these things parallel is by sort of you sort of lay them all out on a grid, and once that's not enough, you put them into computing clusters, and once that's not enough, you put them into warehouses, and the energy requirements, approach those of cities, and we're still only at computing clusters that can run a tiny fraction of the computational complexity of a human brain, which runs on a few watts of energy. So that approach is never going to work. We use it right now for early stage research, and that's fine. But it's not how the futuristic technology of whole brain emulation as a sort of as a commodity industry will work.
So putting your crystal ball on, are you allowed to make speculation what kinds of computation will be required? Yeah.
Well, we have early stage new computing architectures that are not von Neumann. There's nascent work in the last 10 years that has been getting a lot of press in the last 2 years. And this technology is called neuromorphic computing. And some people will cringe. They're going to say, oh, the current neuromorphic chips that are coming out in 2018 would never be able to emulate a brain. And of course not. That's not the point. The point is that they are a completely new kind of design. And they aren't just a little bit better. They are several orders of magnitude more energy efficient. They can achieve certain kinds of computations, and these are neural net-like computations, literally at percent points of the energy utilization of more conventional computers. And this is only in 2018. So I think that these other architectures portend what computers could become, in 100 years. If we design computers that don't work the way we think they have to work because we think a computer is what's on our desktop. If we generalize our idea of what a computer is to these neuromorphic designs, which are essentially physical structures of neural networks. And if we generalize that out and sort of extend that for 100 years and ask ourselves, what would that technology look like? Well, what it will look like is essentially a technological version of a brain. I mean, that's what we're talking about. We're talking about physical structures that have massive network sort of structure to them. And they will be the kinds of computers that we run whole brain emulations on. And we will obviously get there. We have proof of concept that matter is able to be sort of arranged in the necessary way to do this because brains prove it. So the fact that brains exist proves that you can get matter configured in such a way that it can perform these computations with a couple watts of power. So that proves it can be done. Now we just have to come up with the technology to do it. And given time, we will surely get there.
Right. I mean, some people might have an adverse reaction to existing inside of a computer. But
so there's two approaches.
Our brains are considered computers to some people in a sense. I mean, we don't have direct experience of the real world. We have sort of through our perceptions, our brains create models of the real world, and then that's what we actually experience internally. In a sense, our brain is like a virtual, like our brain supports a virtual machine. There's a good enough way for us to sort of utilize and survive.
There's two ways we can approach this. One way is to get into some deep philosophical conversation about whether what you sense is really there, and what we only really all we really know of the world is what our senses give us. So why not put it in a virtual world? I think that that's a perfectly reasonable direction to take the conversation in, but we don't have to. If people are just uncomfortable with the concept of only existing in a virtual world, then there's no particular reason that whole brain emulations can't be further embodied in robotic bodies and just live. And I always like to check what I mean by robotic. A robotic body doesn't have to be rigid and metallic, and in my mind, by the time we're able to build whole brain emulation computers, our robotic technology will have evolved to the point where you won't really be able to tell the difference between what's a robot and what's organic. Science fiction has given us a few examples. The Battlestar Galactica reboot had robots that were essentially human-like, and that has been done in a number of other science fiction examples as well. So when I say a whole brain emulation will be given a robotic body, some people cringe because they don't want to live in something that looks like a garbage can with metal legs. But of course, that's preposterous. That's not what we're talking about. We're talking about lithe, supple, graceful bodies that are as beautiful and limber and capable as biological bodies. And that is what I mean when I say that we could, embody a whole brain emulation such that it can interact in the physical world. So all I'm saying is we don't have to obsess over virtual worlds.
I mean, it's a bit of a dystopia, but Altered Carbon is a television show that shows, mind files are basically put on these little discs that are put into the base of these human bodies that are grown in a lab without proper brains. And so everything that's important is encoded into these little discs or objects that people get placed inside bodies when their original body gets shot or killed or dies for some reason.
That's actually an interesting example. In Altered Carbon, they didn't even describe it as a robotic body that is as organic as a person. Instead, they just said they grow cloned bodies essentially, and these whole brain emulations are basically given biological human bodies. And that's another way of thinking about what the futuristic sort of extension of this hundreds of years, or at least 100 years, could look like. So again, I really want people to not take the word robot, in the unintended way. It shouldn't be taken as C-3PO and things like that. It should be given, the credit it is due. Let the robotic technology evolve in your imagination the same way that the computer that runs the whole brain emulation has to evolve in our imagination in order to get 100 years out, so that we can sort of picture what this might actually consist of.
Well, it's interesting. I mean, like having a look at this sociologically a little bit, I'd like to talk more about the technical stuff, but it seems as though people are more and more becoming sort of accepting of these sorts of ideas, given the amount of media attention to either fantasy or science fiction, speculative fiction, focusing on the what would happen prognostically. Was it Dark Mirror? Has a lot of episodes
Black Mirror.
Has a lot of episodes about uploaded minds being sort of put in a virtual world and then being tortured and all that sort of thing. But I guess the thing is that people are getting people are getting used to the idea that this is a possible future, not the torture, not the the exact implementation,
but the technical feasibility.
The technical feasibility.
And it is interesting. What does that imply about our society? The fact that people are coming on board with this means that everybody's philosophical stance about this seems to be evolving a little bit away from the body identity and toward the pattern identity. Even in the general sort of non-technical population, or population of people who don't think about this unless it's a news article in front of them, everyone seems to be coming around to the idea that pattern identity is probably worth giving some serious consideration to, because we're sort of exposed to it in our science fiction more and more now. So that's interesting that the idea is sort of gaining a certain credibility, although at the same time there's a lot of naysaying about it in other ways. But what were we going to say?
I guess I was going to ask you if anything, given what looks like a direction of evolving ideas in general, people seem to think more and more that it's possibly possible. What would you say outside of a nuclear war or some sort of a devastation which stopped scientific research, which if there was a cataclysm of some description, some environmental disaster, what in terms of philosophical ideas gaining precedence in human society, what sociological problems could sort of turn people around that would stop people from thinking in this general direction, or the evolution of ideas going in this general direction of the computational theory of mind and the possibilities of mind uploading?
Well, it's very hard to falsify or prove or disprove these sort of theories of mind. So I don't think we're going to see some philosophical sort of sudden discovery that renders the whole idea implausible. Because what sort of neuroscience experiment could prove that qualia cannot be, qualia is your sense of red, your sense of taste, your sense of pain? What kind of experiment could prove that a sufficiently complex neural network in a computer can't experience qualia? What could prove that it can is also a good question, but also what could prove that it can't? Because that's to your question. What sort of philosophical experiment would suddenly turn everyone off to the idea, was your question. And my answer is, I think it is hard to prove that you do or don't need some particular aspect of the brain in order to have qualia or consciousness, or I don't even like to talk about proof of these sort of preservation of identity claims, because I think that's more, it's more about just finding a consistent point of view than it is about a proof. If you can find a philosophical position on the nature of identity that makes sense, then that's probably as good as you can do, frankly. I mean, you can't prove that someone's identity is or is not preserved. All you can do is form your worldview around sort of a perspective that is as sensible as possible. Now, aside from kind of philosophical upturnings, what else could?
Just one thing. I mean, like people have brought up the idea that you could, future technology. Hey, that conjured up a lot of possibilities. Maybe we can sort of network our minds in a sense and have an experience of somebody else's experience. But do you think we'll always be having like a personal experience of somebody else's qualia? It still needs to go through this personal sense of filtering, this filter through our own experience in order for us to experience it? Or do you think we can tap directly into it's possible to tap directly into other people's experience in a sense?
We can tap into their neural experiences. So we could, if we can find the mapping between any two brains, if we could figure out how the neural firings in my brain map onto the neural firings of someone else's brain in the presence of similar perceptions. So you show two people the same cat and figure out what part of their brain is firing in a similar way. Then, of course, you could have one of them look at a cat and take the neural firings and translate that over to the other person, and then the other person will have the experience of seeing a cat even though it wasn't there. And this isn't even, I think experiments like this have actually been done, essentially. So we can copy over the neural experiences, but what would be very hard to prove is that you're translating the qualia. So if I see something that is red to me, and we find the underlying neural pattern that is inspiring that, and we put that neural pattern into your brain so that you have some sort of color experience, how do we prove that the color experience you had was red? If anything,
so what you're doing is you're talking about that which inspires the qualia, but if the qualia itself were a pattern and could be reproduced, could you take the pattern of the qualia and have that fed directly into the experience mechanism of another identity?
All we can do is assume that it all makes sense. Right? So we can find the neural pattern that is common when people see something that is red, and which doesn't occur when they see something that is blue. Maybe we will even find that everybody has some similar neural pattern. I mean, for one thing, we know that there is a similarity sort of at the surface. We know that the red photoreceptors are the ones that fire. So at that level of neurology, there actually is a very distinct commonality across multiple people in the presence of this sort of red sensation, which then leads to a red perception. But what would be harder to prove is that your qualitative experience of it is the same. Daniel Dennett, of course, writes about this a lot.
Right. Does Daniel Dennett or you think that experience has very distinct edges? There's a sort of a magisterium of experience that we have that cannot be penetrated empirically at all.
I agree. I think that qualia can't be empirically touched. Sorry?
So that you don't think, I'm wondering if there's any possible direction in philosophy which could treat qualia or experience as having more of a gradient edge to it rather than a distinct boundary.
Some of the popular thought experiments that come up in these discussions are dancing qualia, and what are the other ones? Fluttering qualia. These experiments where you can sort of, and I think Dennett is, again, one of the purveyors of these thought experiments, where you can sort of figure out what the qualitative experiences are that are associated with certain underlying neural patterns, and then you build some sort of, this is a thought experiment, you build some sort of switch that flips back and forth between certain neural experiences and have the person self-report on their qualia. And the question is, are they able to actually have a qualitative experiential difference that they can report, or do they not realize that a difference is happening? I haven't read about these thought experiments in several years, so I'm a little fuzzy on it. But that gets to your point, that the very idea that qualia itself might be a fuzzy concept. It might be so fuzzy that we can't really nail it down. And I certainly think it's very difficult to translate it to another person. The best we can do is say that this person has, let me see, so at an outer level, we can say that these people have similar sort of Skinner-like behaviors in the presence of certain qualia, so that we all react the same way to pain. And then you can, well, actually, that's not true. I personally tend to be more susceptible to pain than others. So people do have different sort of experiences of pain. But there is obviously some sort of behavioral, sort of broad, similar reactive, which obviously is what evolution latched onto in its utilization of pain to sort of protect our bodies from damage. Now, the next question down is, do we have similar neural experiences in the presence of pain? And obviously, we have some similarity there, because we know that sort of similar chemicals are involved. That's why painkiller medications work on multiple people, because the underlying sort of chemistry is similar. But again, is the qualia exactly the same? And I think that the best we can do on this is say that the neural experience is verified to be the same by some sort of neural scan, and the outward behavioral sort of experience by third-party observers. You can say that two people look like they're behaving the same. This is Skinnerism, I think. But you can't really get that layer in between. So we got the neurology, and then it goes through some sort of cognitive qualia experience, which is sort of unreachable, and then that gets translated into outward behavior, which again we can sort of say is similar across people. And I think that layer in between is, I think philosophically we're going to be grappling with it for a while. And the fact that it's hard to get at doesn't mean that we're going to pass it over, because as these technologies mature, people are going to care about this a lot. And I'm not quite sure if we're going to solve it, but we're definitely going to puzzle over it as these technologies come into being.
I certainly agree there. Yeah. So, well, now this brain preservation, the preserving of the pig brain has been achieved, what sort of splashes in the scientific community has this caused so far? What kind of ripples? And where do you see, what do you think will happen in the future? How do you think, after this information has been digested by the larger scientific community, how do you think it will sort of influence the trajectory of research and development in the future?
I don't know. I'm not too tapped into the neuroscience community. I know a number of neuroscientists who sort of are sort of how I learn what's going on in that community through them. We will all sort of watch together to see kind of how academic and professional neuroscience and lab researchers react to this news. And we do want to, we're not just sort of sitting on our hands. We're going to definitely try to take these results, to the community and ask them, we're going to ask them what they think. We're going to ask them, to help us sort of find the parts of it that are, need to be scrutinized further, because that's the sort of rigor we're looking for. Mostly we want to get this conversation going, not only in the general public, but we really want this conversation going, amongst professional neuroscientists. We want them speculating. I think it's generally widely agreed that this preservation is, this ASC procedure, is basically preserving what we believe are the salient features of memory. But that one sentence could be turned into a book if you get into the details. And that, that book's worth of detail is something we want, neuroscientists to really start mulling over, because we want well, not only do we want it to be sort of confirmed, but we want to know that it's definitely doing what we think it's doing. So that's an ongoing conversation that we're hoping is going to begin now in earnest and not let up. As the technology of ASC and other preservation continues to evolve, we hope that the scientific and then the associated societal conversation around it maintains sort of a certain energy and doesn't fall off. Now, that's what I'm hoping comes from this award, is that now this becomes a conversation that we keep having sort of more seriously, perhaps, than we have before.
So aside from achieving the long-term prospect of waking up or reviving a rabbit, a pig, or a human brain inside a virtual world and having it report that it feels conscious and feels like it's an identity in some way or another, the near-term prospects of developing further experiments to, I guess, like to hone our understanding of how preservation, with the outlook of revival into an uploaded form, or even biological revival that on the table at some stage as well. What sort of experiments do you think should be focused on in the near to medium term future, aside from what's already obviously been done over the last couple of experiments?
So you're not asking what the immediate next stages in research are. You're asking what would be the practice of revival?
Yes. No, that's more to what I'm asking. What kinds of existence proofs can we further continue? What research projects do you think, well, for the brain preservation to shepherd and endorse and even do, what sort of projects will be able to be done in the near to medium term future, which maybe also may actually convince more of the scientific community or whatnot, and either somewhat falsify or somewhat endorse the idea that this technology is worth pursuing? What sorts of experiments?
One of the most boring and straightforward and important things to do is reproduction. We don't. One thing we need to do is not actually do the next step of this. We just need other neuroscientists around the world to basically attempt ASC and confirm for themselves, or better yet, have one lab perform ASC and then give the preserved brain to another lab to perform electron microscopy on. This sort of replication of results should be done by as many independent labs as possible. That's a completely uninteresting answer, because it doesn't suggest next generation advances. It is just, that is the careful forward progress of scientific advancement, and that is, in my opinion, that's what I would like to see. I'd like to see a whole pile of labs all around the world say, okay, well, now we're going to try this to see if we can do it. Would be great to see, in my opinion. As for what the next generation research is, I'm not going to prescribe that. I have no idea, but I do think that the BPF, it started out with two major sort of prizes: the small and large prize for a rabbit and a pig. The next step is essentially to sort of do something along similar lines. I'm not entirely sure exactly what that will look like, but the BPF is going to continue to try to motivate ongoing research into brain preservation. The story doesn't end with this award. The story ends when we have a standard medical practice that is cheap and affordable and widely accessible and as commonplace as any number of other medical procedures that we take completely for granted. That's when the story, is sort of ended. At least the sort of R&D part of the story. So a lot of it will sort of maybe pivot toward advocacy to some extent. But again, I don't want it to jump into advocacy and sort of leave all the science at the door. I want to see more scientific validation. It should be attempted on ever larger brains, I presume. Perhaps there are, sort of extended circumstances that can be tested. How a person or an animal dies and you aren't able to get to them immediately. So what is the window of time? Does 5 minutes make a difference? Does 10 make a difference? these are the questions that are going to be important before we can make a standard practice out of it, because we'll have to know what the parameters are of a run-of-the-mill procedure. But I don't know. I'm mostly pretty whimsical about this, because I'll leave it to other people to figure out what the next stages of research are. Yeah, I don't think I'm the right person to design those experiments. I don't know.
Sure. Well, in your opinion, what are some sort of, I guess, other tracks within biology and general science, such as maybe systems biology and general organ preservation for other purposes, do you think will continue, and whose discoveries may feed back into the increasing the viability of whole brain preservation?
Yeah. Well, another area, and this has nothing to do with ASC preservation, but another area that people are looking into is, sometimes they call it hibernation or other forms of stasis, which has a variety of applications. The more fantastical applications are sort of long-term space travel. You could getting to Mars isn't actually so long-term that you couldn't stay awake the whole time, but one possible solution to avoiding the radiation problem would be to go into stasis and then just sort of shield your body under sort of a thick wall or something. I'm somewhat speculating, but using hibernation for space travel. Another sort of much more sort of down-to-earth application of hibernation, or that's not necessarily the right word, but some medical stasis, is either battlefield or like car accident and other, or so car trauma, accident trauma damage is a medical intervention which takes a person who is in a very serious, immediate, life-threatening situation and puts them into some sort of rapid, short-term stasis. Hours or days. Salination. Sorry.
It's just a salination, like where people are sort of pickled in a sense with.
I don't know.
I guess salt-like.
I've learned a little bit. I have learned and then forgotten, because it was a few years ago, what the state of research was on this kind of short-term emergency hibernation or medium-term kind of space travel hibernation. I do know there's a lot of really interesting work here. You can imagine there's a lot of money in it, because the military is very interested in helping soldiers that are wounded on the battlefield. And again, as I said, it has sort of everyday applications. I mean, the number of people who die in accidents and don't die immediately, but are injured in accidents and then over a period of hours, sort of lose their opportunity to be saved. There's an obvious application for figuring out how to essentially preserve a person sort of on short time scales, and then in a way that would revive them later. I'm not actually sure how too closely related that is to the whole ASC and long-term preservation issue, but it's it's all kind of part of the same chapter in some sense.
Okay, well, it's been wonderful speaking to you again, Keith.
Yeah.
And I think also the listeners and viewers who have tuned in and had a big chat in the associated chat room next to the video there.
I've been watching the comments roll in. I never directly steered the conversation to any of the issues that came up, because I couldn't think that fast while I was talking. But
yeah, me too.
it was nice to see some of the interest coming in there. If you want to chat me up about any of this, I hang out on the Carbon Copies Facebook group a lot.
Yep.
The brain preservation also has a Facebook group, by the way. So
yeah, indeed. So, what about for interested, aspiring researchers who might want to find a career in it, or focus some of their existing research? What should they do?
Well, we need we need work all over the place. So, on the sort of medical side, we just need more neuroscience and medical research into how to make this procedure reliable and easy and cheap and sort of everyday. On the computer architecture side, we have to design these next generation computing architectures that can run whole brain emulation. Also on the hardware side of things, we need to advance the technology of slicing and scanning a brain in order to scan out the connectome. On the software side, the actual algorithm for reconstructing a connectome from these slice scans is not a fully solved problem. It is a problem that is has been tackled very heavily. Even Google has gotten into the game because they're so good at image processing now. But it's definitely not a solved problem. There are subtle ways in which reconstructing a connectome from the two-dimensional slices is essentially not solved at that scale. Like, we can do small sections of brain, but we cannot reliably, automatically, without human people putting eyes on it, get the whole thing built. And a brain is so big that it has to be automated. You can't say you need a person to come in and figure out where this synapse went or where that synapse went, because it has to be totally, completely automated. And on other software issues are just sort of, how would you program and create, a whole brain emulation of a very large, of a human scale brain? So there's work for chemists and medical researchers. There's work for EE and computer architecture people. There's work for software. There's work for sort of advocacy in politics. There's work in law. There's piles of work that needs to be figured out in law and regulations. But honestly, anyone with an interest in this can find a way of applying their particular skill set and interest in a way that moves this whole field forward. There's work to be done absolutely everywhere.
Yes, sounds very, very involving and very, very difficult at this stage. But it's certainly an amazing experience. Out of like all the possible research endeavors and things to have a hobby and understanding how the mind works and solving some of the problems, such as continued existence and the ability to live in futures that we may want to live in. It's all like very amazing to be part of. Yep. And to see that there's people working on specific problems to achieve these sorts of things is really great. And hats off to you and at Brain Preservation, Kenneth Hayworth, and also the people at Carbon Copies, Randall, who I've interviewed a number of times as well. It's great to see you guys making progress there and putting your shoulder to the wheel and pushing forward. So yeah, I appreciate all the work you've been doing, and I think a lot of people do. You may not hear it, but it's out there. So keep it up.
One tiny little self-plug. If anyone, the BPF put out, Kenneth wrote a press release, which appeared on several sites and is prominent on the Brain Preservation Foundation's website. Another publication that came out, which is a little bit less sort of press releasey and technical, is I wrote a blog article that was released at the same time, which is available again on the Brain Preservation Foundation's blog page on their website, and it's also available on IEET.org. If you just go there and go to the blog, it's right there. So those are the two. If anyone wants to read a little bit more about this, read the press release that Ken wrote first, and then if you're interested, read my article, Implications of the Prize, which is available on IEET or on Brain Preservation. So I think that would be, if someone wants to get this right from the source. Those are the Brain Preservation Foundations. Yeah. If
you give me those links to the specific articles, I'll provide them in the description so people know exactly where they are.
Yep, I will. Absolutely. So thanks again. It's always a pleasure. I think you've interviewed me several times now, and it's always fun to chat with you.
I always enjoy it. So thank you so much. And you have a great rest of the day, and I'll endeavor to do the same. And thanks to all our viewers. If you like this material, please subscribe, and see you next time. Bye for now.